Thank you for joining the nSolver video tutorial series. This video covers creating basic visualizations in nSolver. In this video, we'll cover the different levels of data created in an experiment and reasons to choose one over another for analysis, initiating a basic analysis, and we'll cover the different visualization options. We'll mainly focus on heat maps, but we'll also touch on scatter plots, box plots, violin plots, histograms, and the available customizing options for each. Open nSolver. You should have already created an experiment. If you missed this step, see our Experiment Wizard video in this series. Your experiment should be visible on the Experiments tab. Expand the navigation tree by clicking on the plus sign to see all levels of data. The raw data contains unprocessed data. This data level is typically used in advanced analysis visualizations. We'll cover this in a forthcoming video in this series. The normalized data contains data after normalization and any background correction you chose to do have been applied. This results in digital, precise, relative counts of genes across samples. Biological interpretation should be done using normalized data. We'll use this level of data for basic analysis in a few moments in this example. The grouped data contains groups as defined by the annotations created in the Experiment Wizard. This level of data can be useful for visualizations such as scatter plots or heat maps in basic analysis. The ratio data contains fold change results and statistical inferences from ratios between these groups. This level of data can be useful in detecting, visualizing, and determining the significance of differentially expressed genes. The analysis level has any analyses that you may have performed. We haven't created any visualizations yet, so let's get started. Select the normalized data under the experiment name, and then select all samples in the central table. Select the Analysis button. This will launch the Analysis Wizard. Specify a unique name for the analysis, and under the analysis type, you'll see that you have a number of different options. Agglomerative clustering creates a heat map. This is good generally for seeing how data clusters. You may find genes that cluster together, indicating that they may be part of a common pathway. Samples that cluster together may be part of the same group, or may be technical or biological replicates. The patterns you observe in heat maps may give you an indication of heterogeneity or the presence of confounding variables or outlier samples in your data. Violin plots and box plots are each useful in displaying the distribution of counts for a single gene. Scatter plots can depict reproducibility between replicate samples, can be useful in identifying outliers, and when used on group-level data, may highlight potentially differentially expressed genes. Histogram plots show the distribution of data by probes and by samples. We'll start by selecting agglomerative clustering. The checkboxes below this field allow you to exclude samples and information that are not pertinent to your analysis. The optional histogram filter will enable a tool for automatic filtering of samples or probes used in the analysis. This can be especially useful for larger data sets where manual sample and gene selection is more time-consuming. Checking the next box will exclude any samples with QC or normalization flags from analysis. Remember, however, that flags simply indicate data that should be checked a little closer. Nanostring recommends reviewing your data before deciding to exclude any samples from analysis. The next box will exclude any control probes from analysis, and you can also choose to exclude any content normalization probes, which are ones you selected as housekeepers and felt were expressed stably enough to use them for code set content normalization earlier in the experiment creation process. Select Next. If the histogram filter was enabled, the next window has a number of customizing options. Along the top, you can fine-tune the filter granularity by changing the number or size of the bins. By default, outlier samples or probes which fall outside the filter range will be excluded from the analysis, but checking the inverse selection box will reverse this selection. Note that the plot on the left filters samples, while the plot on the right filters probes. Within each plot, we can drag the pink curtains to manually select what we are keeping in white and what we are excluding from analysis in pink. Below the plots are drop-downs where we can set the lower and upper bounds of our selected area with specific values. For this example, we'll filter out all probes with counts 20 and lower in the probe filtering plot. Either drag the pink filter from the left or type 20 in the lower bounds box under the probe filtering histogram, and we'll leave the sample filter plot alone. Select Next. The next window shows us the samples that will be included in the analysis in the Selected Samples field, after any filters we may have applied from the histogram filtering screen. We've chosen not to exclude any samples, so all 12 should already be in the Selected Samples window. Any samples that appear in red indicate flagged samples. Select Next. The next window shows us the probes that will be included in the analysis in the Selected Probes field, given the selections that we made in the previous windows. Most endogenous genes and proteins should already be in the Selected window, unless they fell below 20 counts. And all control genes should be in the Excluded window. We can change any excluded probes now by selecting them and moving them to the Select Probes field with the arrow. We can also do additional sorting and filtering here by using the column headers. For example, instead of applying the histogram-based filter earlier, we could have implemented the same probe removal by clicking on the average count header to sort by average count, and then exclude all probes below a certain threshold. Select Next. The following window is customizing options for heat maps. Most heat map settings can be left in the default mode. Choose Z-score for genes if you would like to see a heat map depicting each gene's relative expression across samples. Choose Z-score for samples if you would like to see genes' relative expression levels within a single sample. Euclidean is the most sensitive method, which may be helpful in some datasets but is not ideal for datasets that may have outliers. Clustering determines the distance between things. Leave on if seeing data for the first time, since this can give you a good first general impression of your data. Under Select Sample Annotation, each annotation in your experiment, treatment in this case, as well as treatment group should be checked. If sample names have been populated, it may also be helpful to uncheck the file name box, since these long names may make visualizations more difficult. Under the Select Probe Annotations, gene name and protein name should be checked. Select Finish. A Java window will appear with the heat map. Use the slide bar to view the names of the samples in columns at the top. Adjust the label settings if needed. Select View, then Labels, and change Rows to Name or YORF to view the gene names in rows on the left side. Adjust the label font and the size, and use the Keep Fixed checkbox to fix the column names in these settings. You can also change the colors of the heat map here. Note that the samples have clustered by treatment type, unstim on the left, stim on the right. Note also that we can scroll through the probes in the area with the decreased expression, red bars, or the increased expression, green bars, in the stim samples. Also note that sample 11 seems to cluster slightly differently from the others. These are the types of patterns you'll want to be aware of in your data. Next, we'll briefly talk about making a Violin plot. To do this, we would go back to the nSolver dashboard and do many of the same steps over as before, selecting normalized data, analysis, only we'll select Violin plot from the list in the analysis wizard. When we selected Finish, we would see this screen. Selecting lines in the Genes field displays results from these probes in the plot. We can search for probes of interest above in the Data Filtering field and can use the Show by Condition box to group results by our annotation, in this case treatment, and overlay the results from the selected probes. The File menu allows you to save the image, save settings, or print, and the Settings menu allows you to customize settings. The process for creating box plots is nearly identical to that for Violin plots, so we won't detail that here. Instead, we'll touch on scatter plots. Selecting Scatter Plot from the Analysis Wizard list and selecting Next prompts us to select the samples we want to see in the scatter plot, and select Finish. The resulting plot has plotted the first sample in the list, number 1, on the x-axis. We can change this by using the drop-down at the bottom of the screen. This sample's results are plotted versus all the others we selected. We can drop some of them from analysis by unselecting the checkboxes next to them. The File menu allows you to save the image, save settings, or print, and the Settings menu allows you to customize settings. Lastly, if we select Histogram Plot from the Analysis Wizard list, we'll see a histogram that represents all the probes. Selecting anything in the Genes field will change the histogram to reflect the data only from that probe or probes. We can search for probes of interest above in the Data Filtering field. We can fine-tune how the plot looks by changing the number of bins, or bars, or by changing the bin size, which is in log2 counts, since that is the x-axis metric. The File menu allows you to save the image, save settings, or print, and the Settings menu allows you to customize settings. Returning to the nSolver dashboard, we can see that all the analyses we created have been automatically saved under the analysis level. We can return to this level to access these analyses at any time. This concludes the nSolver video tutorial series lesson, nSolver Basic Analysis Visualizations. You can always contact Nanostring support and check out our other video training curriculum online. Thank you.